Loft, which is our virtual.
We're gonna wait 3 minutes. I'm gonna stand up here awkwardly.
Can you juggle or anything?
I can't dance. I certainly can't sing.
I can juggle, but I don't have any. Oh yeah, that would go real well. I've never juggled before. Just watch them.
We'll see how well I can talk without moving around. I think that's going to be the challenge for all of us. I know. Yeah, and we're doing WebEx rather than Teams. So you guys can't record Teams? Was that the issue?
Yeah.
Why is that? Are we good? Wait for the music to die down. Well, hello everyone, and welcome to today's seminar on advancing research through PMedIC. I'm Bobby Jo Webb Robertson. I'm the Division Director of Biological Sciences at Pacific Northwest National Laboratory. And I've had the pleasure of leading PMedIC for the last 2.5, 3 years. And I am joined by my colleague from OHSU, Beth Habecker, who will introduce herself here in a little while. So I wanted to start out, this is really a history of PMedIC as well as currently where we stand right now. So OHSU and PNNL have been collaborating for many years, before 2016. But in 2016, we had a couple of large programs, 2016, 2017, between the Proteogenomic Translational Research Center and the Undiagnosed Disease Research Network. And so those really sort of spurred a lot of new innovation between the two groups. In 2018, Karen Rodlin, who's here in the audience with us, PMedIC was really her brainchild. You know, she noted how much great science we could get done if we could partner the great technologies at PNNL with the needs of the biomedical community here at OHSU. And so she worked with Aditi Martin and pitched PMedIC as a partnership between the two. Partnership being that both institutes would dedicate some resources to enable building more programs between the two institutes. And both OHSU and PNL have done that. They've dedicated time for staff, such as myself, and as well as seed money for seedling projects and things like that. So that's really where it started. And in 2018, we had 3 projects together. And in 2022, we had 24 projects together. So it really has just ballooned. And in a little bit, I'll explain why PMedIC has worked so well. Laura Erker joined the team along the way. I'm hoping Laura is online. She couldn't join us today, but she's been highly involved and an invaluable asset to PMedIC. And in 2020, Karen stepped down to pursue her next phase of her career. And myself and Beth took over the helm as the co-directors pretty much at the same time. I was like maybe 3 or 4 months before Beth. And Liz joined Laura also as our support on the OHSU side in strategic partnerships. And so we've really been working as a team, you know, connecting people. And that's one thing that makes PMedIC really special and why we've been able to transition co-directors and still continue with the success is that PMedIC's never been about Karen or about me or Beth or anybody else. It's been about bringing PIs together and growing them. So, you know, it's not reliant on one person as that figurehead of we're just going to build, you know, the biggest cancer program we can. It's about diversification and growing programs. And we're going to talk a little bit about some of those. You know, the obligatory vision and mission statement. But really, the core of this is that what I had brought up earlier, the ability to integrate sort of the technology and computational capabilities at PNNL with the biomedical needs and computational technologies and other capabilities here at OHSU to really do some innovative research programs. And that's worked very well, as you could see by the numbers. So what do the PMedIC co-director and broader teams do, right? So I'm not showing everybody's picture because there's too many people, but you know, at PNNL, we also have a team of folks who help make connections and grow programs. And so that's why it was very easy to find new co-directors because we have so many people involved. But what we do is we do identify these networking opportunities to bring people together, right? What's your need? What's your technology? You know, who's looking for what and how can we really drive some new science? We coordinate joint institutional activities like the one you're in today. And we follow and track all the metrics for, you know, how this grows. And we just facilitate joint appointments and all the things that we can do to help build new relationships. A few pictures that was from last year at our PMedIC retreat that we held at PNNL. Laura with Sanjay on a tour of his lab when we were down here. And then I love this picture of Beth and she lets me use it, which I like even more. Shows we have skin in the game. This was a project that we had started looking at tears, proteomic tears to understand pain. And so Beth went in and actually went through the procedure of how they collect these. So we've got lots of great stories. I have a picture I sometimes show my son with a Bass Pro Shop hat that we left here on a family trip, and Beth went and picked it up for us so that he could get his hat back. But going back to PMedIC, and I am a statistician by training. For those of you who don't know me, I've been in computational biology my whole career, but statistician by training. So I'm always a person who gives the numbers on the presentations because that's the fun part for me. But I said we have 24 projects to date. The majority of them are NIH, as you can see up here on the slide. Although we have had several foundation grants. It sounds like we just got a new one, I think, John, it sounds like, with Ruth. And we have one DoD grant as well. So part of what we've been doing is trying to find more opportunities in other spaces. NIH is wonderful, but where can we find even more opportunities to work together? And that's sort of how the numbers break down in funding for each of these. So the center grant, I'm not going to tell the story, but you should go back and listen to one of our prior recordings with Karen talks about selling the center, that James Evans at PNNL was trying to build up our cryo-EM capability. And Karen walked into an office here and they were like, oh, so we really like to go after this, but we don't have the right partner. And Karen's like, I know the right partner. And we sold a $50+ million center. And that center is working on its renewal right now. So it's been going for a while. This comes back to the previous comment that I made. PMedIC is actually held up at PNNL as the model for joint institutes. And I say that because it can grow a lot because we're not centered around a single person having to drive all the science. And so the funding's gone up, but if you look at the little line graph over there, you'll see we've got a lot of programs in infectious disease and cancer and cardiovascular and so forth and so on. And each of those little circles is a different OHSU PI. So as I mentioned, it's a collective of people working on the program. Another point with PMedIC that I think has been really great in recent years. So the green bars here are the cryo-EM center, which is really, made our numbers look really great. We love the cryo-EM center because it's a really big sale, but those are rare. So, we can't just say we're going to just sell more centers. So what we have to do is sell more, R01 scale programs or program grants. And that's what we've done. And so the fact that 21 and 24 or 22 there, you see the green and the black are catching up to each other. And that's because of that big increase in research project grants, which has been really great. The other thing is just the number of people involved. So this graph shows how many staff, and this is not all people involved, these are just PIs. So the number of people actually involved is much larger than this. But if you look at the number of PIs, how they've grown between OHSU and PNNL over the years, means we're adding more people, not just writing more grants with the same people, which is expanding the overall research portfolio. And with that, I think Beth is going to finish the rest of the presentation and talk about some of the science.
Thanks, Bobby Jo. As she said, oh. I'm Beth Habecker, and it's been my pleasure for the last several years to work with Bobby Jo and get to know a bunch of other folks at PNNL as part of this process. And I'm actually now one of the dots on that thing because I have my own collaboration. So along with the research, part of what we've been trying to do over the last few years is start getting the word out more broadly. And so starting these quarterly seminars was one of the things that we did. And this event today is kind of taking the place of one of those seminars. But these are all ongoing collaborations that had reached a point where there was some interesting stuff for folks to talk about. And, when COVID got us all out of our mindset of a seminar is where somebody goes someplace and talks to a bunch of people in a room, and all this stuff was going on online anyway, we said, well, heck, we could have people do joint seminars, and the PNNL person can be in Richland and the OHSU person can be in Portland, and everybody can just log in online, and it actually worked great, and the attendance of those was quite good, and we plan to continue those in the future. The other thing I'll just point out is you'll notice on the far left, the template is different. And the PNNL folks asked us, the communications staff said, hey, would you guys like your own logo and letterhead? And we said, okay. And so, so they made a bunch of stuff and went through a few things. And now there's a separate kind of, and it's all part of the trying to raise the visibility of this kind of joint institute around. So along with all those new grants that you heard about, they're now resulting in a lot of publications and ongoing publications. And Laura has done some work drilling into kind of metrics, and really the joint publications are able to get into journals that on average have higher rankings than journals if you just look at all the aggregate publications from the institution separately. And I don't want to push that too hard because we all have reactive bad thoughts about impact factors and all those things. But just to say, to make the point, one way of trying to quantify for the overlords who always want numbers, that in fact, having people work together is providing opportunities to do new and creative things that are really unique in our high impact. And one of the spaces where that has shown up is in COVID. So Jennifer's here today. Thanks for making the trip. And I think Vikadu's going to be around later. They already had an ongoing collaboration on Zika virus and some other things. And so when COVID hit, they were in a great position to do some really interesting work on how these viruses hijack and change the lipids in human cells. One of the other things we've been working on is clearing a path for students and postdocs from OHSU to be able to train at PNNL. Another thing that, you know, COVID obviously did not facilitate that, but we now have had several OHSU students do internships at PNNL. We have some more of those happening this summer. The dean's office has come up with a little bit of money for situations where people need a little bit of help with housing. And we've worked with the provost and, you know, kind of all of the Byzantine things that have to happen for students to go someplace and get credit, and have been able to kind of lay a pathway for that to happen now. And the other thing that Bobby Joe alluded to is both institutions have put money into developing these partnerships. And really, the money has been for some salary support, but also significant money over several years to fund pilot projects. So for PNNL, that money is called LDRD, and those are internal funds to help support their investigators and pay for instrument time for these pilot projects. And then on our side, we have, I'm not going to remember what ERS stands for, but there are, and it's not important, but the smaller awards, and then also the last couple of years, a couple of $50,000 pilot grants for OHSU investigators to form new collaborations with PNNL. And those things have worked in order to expand new collaborations. And we've got pilot grant holders that are only have a small amount of overlap with the folks who already have joint grants together. So this is really working well to generate new collaborations. So there's a little bit of overlap between the two, but we have been successful at identifying new people and new collaborations. And so that's kind of where we are now. We want to thank everybody. Bobby Joe and I would like to thank everyone for their support during our time as co-directors. And part of what we get to do today is announce the new co-directors. So they, so for the OHSU side, I'll go ahead and go to this slide. So Jamie Lowe, who is an associate professor in OBGYN and has a lab at the Primate Center and probably has other appointments that I'm not saying, but those are the key ones, is going to take over as the OHSU co-director. And she got in here from the Primate Center today in time for lunch. Welcome, Jamie. And Bobby Joe will introduce the new PNNL co-director.
Yep, thank you. So Dr. Josh Atkins is taking over as the PNNL co-director. And Josh has been at the lab for a little bit longer than me, so a little over 20 years. He's a lab fellow, PhD in biochemistry. He has multiple NIH programs of his own. And for many of you here, know him from the omics capability at PNNL. He ran our integrative omics group for over a decade. So welcome, Josh and Jamie. We're very excited to see you guys are going to go next with PMedIC.
All right, yeah. And I think that's it from us.
It is.
And we will move on to the next phase. Whoever's
I think
John?
Josh and Jamie.
Oh, yeah.
I guess I'm gonna come up and introduce John.
And you got to figure out what's the right presentation. Is it this one?
Was it this one that you just did?
That's when we ended.
Oh, okay.
Is
Yeah,
John
that would be
speaking
that one.
first?
Yep.
Okay.
And then, all right. So, yes, thank you, Beth and Bobby Joe, for introducing Jamie and myself. I just wanted to point out that you both are wearing what I would consider fall colors, and Jamie and I are wearing spring colors. So I think it's somewhat fitting. It's my opportunity to introduce John Jacobs. John and I have worked together for almost as long as Bobby Joe and I. Pretty close. He was somebody who was really involved in some of the early plasma proteomics work and is currently our BTRR co-director and director of a pain center grant at PNNL, but has been doing omics work for 20 years as have I. So, John, turn it over to you.
Thank you. Okay, so I've been tasked with talking a little bit about PNL's capabilities and how they can be utilized in collaborative interactions with OHSU. And so if we talk about capabilities, so a few caveats. Number one, this is not an inclusive list. This is actually possibly a highlight of possibilities that are available. So number one, I'll try to put names associated with various capabilities. So I'm just giving highlights here. I'm not giving very much in-depth information, and so the idea is that you go and you ask and dig a little bit deeper with those that have much more information about various capabilities. Secondly, capabilities, if we think about capabilities, you now are implying that there's been some technology that has been developed, or maybe is still in development, and is at a various stage of maturing, and so, which has its own very unique qualities with that. We spend a lot of time developing technologies at PNL. Actually, that's one of our main drivers. That's kind of what makes us somewhat unique. And so I want to talk maybe a little bit about that. And so if we, I want to use a test case scenario here of, if anyone remembers back in the day, you can call it the whatever, the global proteomics panacea. So when I first started at the lab, proteomics was just really getting its footprint. And a lot of us thought that this was going to, well, we did. It was going to revolutionize, biomedical research and research in general. And so one of the earlier slides that we always used to put is you've got these very archaic Department of Genetics and Biochemistry doing these one-off fishing expeditions. And we were going to come in and sweep up and pretty much be able to quantify and identify and do everything except just do it in just much larger quantities and just pretty much make the 1Z, 2Z type research obsolete. So, well, quickly learned that that wasn't the case. It's very important to do 1Z, 2Z experiments. It's still very important. We actually, mass spec is actually, I'll talk about how it's actually migrated a little bit to that area and away from, we talked about broad global proteomics and down to more targeted proteomics. But anyways, this kind of then brought me to an interesting, maybe some of you have been involved in these type of discussions. So there's a situation, if you remember, if any of you know of your classic literature and you think about the Christmas Carol, you think about there's this interaction between, if you remember Ebenezer Scrooge, he's kind of the grouchy old overlord, and Bob Cratchit is his employee. And if you can think of kind of an interaction, maybe you can think about someone who's developing technology and someone who's using technology. You say, back in the day, we used to say, well, here's your list of 400 some odd high abundant proteins which we detected in your sample. And it's like, well, your collaborator goes, well, but I'm not interested in ribosomal proteins. I want to know, where are all my EGFR pathway members? And we'd reply saying, bah humbug, this is great proteomics data. It's even quantified. You should be thankful for all this access to new technology. And then the collaborator goes, okay. And collaborator walks away thinking, wow, this new technology isn't really that great. It's not giving me what I need. And us technologists walk away like, no one's really appreciative of what we're doing here. This is really great stuff. And so if you think about technology, you think of this this graph where you got maybe a trigger that happens, and inevitably there is this peak of inflated expectations. You feel like this is going to revolutionize the world. But inevitably you do get this kind of, call it different versions of it, the trough of disillusionment, meaning that expectations are kind of being not met. Maybe the technology isn't quite mature yet. It hasn't really found its niche yet. It's working through. But sometimes that takes time. And as you work your way up and you learn a little bit more and you learn about really the impact of specific technologies and where it can hit, you find this productivity plateau. And where that fits as far as when it's available as a capability, I won't say where any of our technologies maybe fall in this realm here. Hopefully, by the time we feel like it's a capability that we actually feel like it's going to be useful to the applicants and going to be able to be drivable. But we are driving technology, and that's what we like to do. We like to keep on the cutting edge of this, and we like to keep moving forward. And so we also like to manage expectations with with those that we collaborate with. So, anyways. So this is just specifically for proteomics. I mean, you've got a various number of challenges, reproducibility, throughput, that are all interrelated, and sometimes there's trade-offs of either one. But then also there's a tremendous amount of strengths that as we work, specific for proteomics, as we work through this, trying to find the unique and the most impact for what's been developed over time. As far as what we're doing here, you can kind of divide our break down the interactions. You think about applications, biological applications as one component, but it is interrelated to a lot of other, as a capability, it's interrelated into like mass spec and imaging, hardcore technologies, instrumentation, on the floor instruments. You got the chemistry and biochemistry components. You can get, I'll talk very briefly about activities and PTM methodologies. And then it's all tied together with computation and how that interrelates to the data that's being generated and actually the flow of data through from the beginning, the sample prep, all the way till, you know, the end of the data when you actually have to make it interpretable. And so I'll kind of work my way through some of these. Like I said, this will be a very high-level review of a lot of different technologies. And if there's anything that you don't see that you're interested in, feel free to ping us. But I think the easiest one to start with is the PNCC. It's already been highlighted. Pacific Northwest Cryo-EM Center. James Evans, Eric Guo, and Steve Reichow. It's OHSU PNL Joint Center. Been highly successful. They said it's in its renewal stage right now. You just look at the numbers that that up until, you know, just this last month, the hundreds to thousands of hours and projects that it's been associated with and has been generating. What I like is I like the percentage of awarded compared to the submitted. It's 92%. You know, if you're interested in it, they'll probably get you involved and get you, get working with you. And obviously it's been highly productive. The amount of structures that's been deposited in PDP and yeah. And it also has a tremendous training mechanism as well. It varies from, a remote screening, remote training with students for multiple days, moving on into, more hands-on, on-site sample prep work, to actual, microscope operation, and then longer-term type internships. I don't know if James is online, but obviously James Evans would be the contact at the P&L side for anything PNCC related. If we go into the mass spec side, which is kind of where I reside primarily, you think of it, global mass spec, global proteomics is kind of the workhorse foundation core that a lot of the capabilities then devolve from and expand from. And so this still remains a very common, a very driven type analytical platform that we provide. We still continue to focus on increasing sensitivity, increasing throughput, and increasing reproducibility. We've got versions of this that depend upon the system that you're interested in. Talking a little bit about, we have refined for many, many, many years how we can quantify and dig deep into plasma, biofluids, anything biochemical, anything biologically oriented, cell culture systems, you name it. It's just kind of the bread and butter mass spec platforms that we use, and then everything kind of divides from that. Thinking about some of the most recent capabilities that have come to play, nanoscale. So we call this, the deepest dive is called nanopots. Pretty much stands for one pot type of analysis. We realized that if you wanted to get higher sensitivity, you need to get rid of manipulations and keep it as simple as possible. Only way we could do that is to do it all in one single pot and then take that and then inject that onto the mass spec. As its name implies, we have nanoscale. So you're thinking about the amount of protein that is required to be able to get a reasonable coverage. We have nanopots. We also have micropots, microscale. And we've essentially developed platforms associated with each of multiple scales of needs for whatever particular biological system that you need in-depth proteomic analysis with. This has also been coupled with flow cytometry and we're driving towards single cell analysis. I mean, so to be able to match that with what's going on the genomic side. Also, we link it very efficiently with laser capture microdissection. So being able to capture down to about 50 micrometer scale for spatial resolution, being able to capture, getting close to single cell. But this is an image that we use for, we have its collaboration with Jake Estes over at VGTI. And in looking at capture of neighborhood, they're able to provide images of SIV infected individual cells. And we're going in and trying to capture the neighborhood around an infected cell versus a non-infected cell. And that's going to be it's a very powerful technique in conjunction with the very single cell level resolution they're able to provide on their side. For targeted mass spec, so talking about global mass spec, now we're narrowing it down to say we really only want to look at a very select number of targets and be able to identify and accurately quantify those particular targets. It's somewhat of a similar but yet a distinct platform that we use. Generally, it's a triple quad mass spec where we go in and we actually, as the name implies, we're targeting we have targets, which means we have to have that predetermined, which means we're actually spiking in peptide standards. So this is an antibody-free system. All we need to do is know a sequence. It can be any sequence. And that's the strength of something like this, is that as long as we have a peptide that we know we can visualize in the mass spec and is quantifiable, we can heavy label synthesize those peptides and we can multiplex that up to a certain quite a large scale and be able to accurately quantify, that particular sequence, which then we can infer our protein quantification. I like to show this. This is just a protein TREM-L1 that we analyzed. The power of this is, number one, you're not worried about affinities for antibodies that are typical for ELISAs or any type of platforms like that. Since it's sequence specific, then it can be essentially agnostic to species and it can be agnostic to where on the protein it resides. And so we had this fairly complex protein, TREM-L1, has an extracellular, has a transmembrane, and has a cytoplasmic domain. In some species, the extracellular domain is expressed. In some species, the cytoplasmic domain is expressed, and they all have different sequences. Trying to capture across species quantification of this protein in all its different domains was probably virtually impossible using any sort of affinity type technology, but it was actually very straightforward with SRM, where you could just go in and we just had specific sequences we could quantify for each of these different areas, and we could very quickly, and it's actually in a single assay, develop a TREM-L1 cross-species assay that we knew the quantification of it and which part of the protein was being expressed. So along the same lines, we've developed some very sensitive techniques in order to dig deeper into, for targeted mass spec, we call this PRISM. We also have other approaches, long SRM and PRISM plus longer SRM and other different techniques. Pretty much allow us for multiplexing up to, this is about 150 proteins. We can multiplex up to that amount. But the idea is that we're able to dig, with the platforms like this, we can be just as competitive with your most sensitive ELISA assays. There's throughput when you get down to that sensitivity, I mean, there's trade-offs with throughput, but for the most part, if you feel like you can see it at a low picogram or type level, we can probably detect it using the approaches that we have for targeted. Now, I didn't really talk much about PTMs as all, but any of these approaches can be utilized to go after various PTMs. The most common one is phosphorylation. And so you can do global phosphoproteomics. That's pretty much standard approach for us. We generally do it in conjunction with a global proteomics, so you can actually get a protein quantification along with a specific phosphorylation. And then we're actually getting down to nanoscale phosphoproteomics as well. We're able, to be able to capture, whatever near single cell type phosphoproteomics and being able to have that difference in signaling mechanisms being able to be captured. On staying with the mass spec side, looking at metabolomics and lipidomics workflows. So Jennifer Kyle, she's here. Any questions, you can direct it toward her. And then Ernesto, right there as well. You've got, we both have LCM chromatography for metabolites, GCMS for metabolites, depending upon their separation characteristics, and then also we have a very high robust LCMS pipeline for lipid analysis and data analysis, and you can actually do all of it all at once if you want to with various separation techniques that we've developed here at the lab. So John asked me, he said, hey, John Melkor, he's here. He said, I hear you're giving a talk about capability. I said, can you throw some slides up for me about some of the things we've been doing? I'm like, yeah, sure, you bet. So if you have any questions about this, you can talk to John. He is linking, just using mass spec approaches, protein abundance with protein structure, and doing manipulations prior to mass spec analysis that is able to tease out protein structure, and hence, if those who are involved in protein structure know, structure denotes function. So protein function, can change based upon, structural elements of what's happening. Other than, generally proteomics, we're not so worried about structure itself. We're just worried about its overall abundance, but it's structured based upon PTMs and other binding characteristics can be altered. And so there's a couple of techniques that he's been linking. So along with protein-protein interactions, post-translational modifications, other drug modifications, structure can change, which then will alter the function of the proteome. And so by doing a couple of upfront techniques and separation approaches like thermal proteomic profiling, changing and isolating various denaturing, denatured products, protein products, and limited proteolysis, and looking for kind of the blind spots during looking at different conditions, you're able to then tease out certain structural aspects functional structural aspects of the proteome. And he's done John's done some great examples in the diurnal cyanobacterial light dark cycle with glutathione reductase and thermal denaturizations, and also with in an infected model system in COVID, being able to identify RNA binding and zinc finger binding regions of proteins. And so I'll let him give you any details about that later on. So one aspect that's a little more advanced is something our lab's been working on for a while, is called SLIM. Some of you might have heard about it. It's structures for lossless ion manipulations. And so it's really, and taking, if you've heard of ion mobility spectroscopy, so it's the ability to separate ions that are essentially identical in mass, but have, that are isobaric, but have different structures. You can think about metabolites that are identical, but yet are different isoforms, trans versus cis, or even at the protein level, you can think of phosphorylation events. You've got maybe a peptide you've isolated, and you have a phosphorylation either, at this serine here or this serine here. You can't resolve it, but it's identical mass, but you need to be able to separate that or resolve it. And so taking as the SLIM approach is essentially what we're doing is we're taking this ion mobility separations, it's getting put on a circuit board. And by able to do that, we actually are able to almost make an almost an infinity length type of an isolation. So being able to isolate out structural components that are essentially inseparable previously. And so, I mean, example of this would be, an L versus D conformation of an amino acid, or a cis versus trans type of a phospholipid, or any sort of structural difference of a molecule that has an identical mass. Some of the projects I'm associated with have to do with trying to determine and elucidate complex phosphorylated regions of proteins and being able to isolate those out and be able to be accurately determine which site is phosphorylated, and provide a somewhat of a fingerprint or a blueprint for what's affecting downstream signaling. I know there's 3 phosphorylations occurring at a particular site. I just don't know which direction, which phosphorylation sites are actually active. Things like that. This is a very powerful separation technique that is put in conjunction with our current mass spec approaches. Finally, I'll talk just very briefly about chemical biology that we have in the lab. So Vivian Lin, she's here, right there. Any questions need to go to her. We have a very robust and a very active chemical biology group at PNL. It ranges from really across the board. You're looking at activity-based proteomic profiling, and we call it ABPP, where we're looking for protein function studies, developing photoaffinity probes for labeling POW, and also developing fluorescent probes for imaging, cell sorting, and anything along those lines. And these chemical biology approaches really integrate well with other capabilities that's going on at PNL. And anyways, visit with her, and she can tell you more about that. And that is all I've got. I'll stop here. Thank you very much.
Okay, I'm going to introduce our next speaker, who is Dr. Puchalowska, who is the Director of Immune Monitoring and Cancer Omics here at the Knight Cancer Institute at OHSU. Her research focuses largely on the role of cross-reactive microbial antigens and how they may impact the efficacy of immunotherapy treatments in cancer. And also she is involved in many clinical trials, especially kind of leading the research endpoints, connecting researchers and clinicians and making sure that they achieve the research and clinical goals. So I'm here to welcome her today. Let me see if I can pull up your talk. There we go. There we go. Thank you.
Hi, everyone. I'm super happy to be here. Thanks for the invitation. I have a couple of relationships with the PMedIC group so far, so I'm really excited to be here and talk about them. And I want to specifically thank Liz as well as Laura for thinking of me and inviting me to meet you all in person and give you this short talk. So IMCO is what we call our Immune Monitoring Cancer Omics Services. We are located within the Knight Diagnostic Labs right at Markham Plaza. So not on the waterfront and not up on the hill, but somewhere in between. There's no good way to get to us. There's no tram, so you'd have to actually take a car. And we partner and work very closely together with our BioLibrary, which is our human tissue repository, and HSR, which is our histopathology resource courses that help us process all of our clinical trial samples. So what exactly is IMCO? So IMCO really was born out of the need to provide seamless support and comprehensive analytics, specifically in the context of clinical trials and translational trials. So some of our goals include comprehensive characterization of immune responses, looking at mechanism of resistance and activity, as well as developing new biomarkers to predict therapeutic outcomes as well as toxicities. We don't necessarily develop our assays like our friends at PNNL, but what we try to do, we try to take them from the smart people and build some efficiencies or rigors around them to be able to deploy them in the context of clinical trials and clinical samples. So we have a number of state-of-the-art assays we think of as cutting technology, and we try to power them with robust analytics, specifically in the context of clinical care and standards of care. So what does that mean to us? What does seamless support and comprehensive analytics mean to us? So generally speaking, when we think about the pre-analytical process, we think about all the steps that take place before someone presents you a piece of data in a PowerPoint or in a paper. And so for us, that means really taking care of every step of that journey that sample takes from the bedside from the bedside where the patient is donating these samples to us, and then building and maintaining that chain of custody in order for us to really make sure that all the samples are processed in the same very specialized way, ultimately then employing those samples in assays that are informative in a given setting, and then generating reports back to our customers or clients or collaborators. So as you can see here, we make use of the Knight Diagnostic Labs that processes diagnostic samples. Our samples are coming through the same door that our diagnostic samples are accessioned. They're treated in the same way. They get received there, accessioned, numbered, de-identified, they get barcoded, and then they get tracked in our BEMS system. Then they move through course like HSR that I mentioned for processing. They undergo rigorous pathology review, which is done by our internal diagnostic pathologists. And then they come to us for the actual processing and the analysis and report building. So currently we have a number of assays that we offer in this format. On the left-hand side, you have assays that are designated research only. On the right-hand side, you have assays that are designated CLIA CAP certified. The difference between those two, if you are not familiar, is the fact that our CAP CLIA assays are deployed in our diagnostic setting. They can actually inform patient care. The research assays are usually correlatives and other biomarkers that are incorporated into the clinical trial, but don't necessarily immediately affect patient care. So on the left-hand side, we have a number of assays, and I'll give you a brief description of all of them. They include our multiplex immunohistochemistry platform, as well as cyclic IF platform driven by our Luniform machine. We have our peripheral immune contexture monitoring platform, which is called CyTOF, nanostring genomics, which is also a spatial proteomics platform, although not single-cell resolution like multiplex and cyclic IF. TCR-seq, transcriptome, as well as serum cytokine assays that are powered by the Olink technology. On the right-hand side, we have a number of assays that are deployed in a diagnostic setting, including targeted DNA panels for solid tumors as well as heme malignancies, full transcriptomes, nanostring, as well as clinical IHC and FISH assays. So not to belabor this concept, but we spend a lot of time thinking about this organization, and IMCO is involved in every single step of this process, meaning that when a clinical trial is getting designed, we work very closely with our physicians as well as our clinical organizations to make sure that we're build operations into this process in a way that really preserves the biospecimens that we receive from our patients in an appropriate way. We run the diagnostic assay as well as the research assays, and then work with our investigators to really drive the biology and the hypotheses that were addressed in the given clinical trial. So who are our clients? We work on active clinical trials as well as on retrospective trials. We have sponsored agreements with industry partners as well as IITs, which are internal to our clinicians. Once in a while, we'll provide CRO services, meaning that we'll just collect samples from clinical trials that are sometimes across the country or even international, and they come to us and we process them and receive them and store appropriately. We work with BioLibrary for sample collections and again, making sure that everything is stored appropriately. Majority of our trials are in an earlier side of drug development, so some preclinical, but most of our trials are phase 1 trials, first in human trials. 20 to 30% of our work will include assay development or building something new, something that that maybe there isn't an assay and that there's an unmet need to address it. We have a list of our current projects on the left-hand side. A lot of our internal investigators use our labs. We also have other academic collaborators across many different institutions, including Stanford and UCLA, Albert Einstein, and UPenn. And we also have a number of industry partners that we work with as outside industry-sponsored trials. So currently, we run 4 major assays that I feel like are our bread and butter. We have our MIHC assay, CyTOF, our DSP genomics platform, as well as Olink serum cytokine technology. You can kind of see where we are in terms of our annual throughput. Some of these assays have a quicker turnaround time, and that could be potentially seen by Olink. We have processed almost 4,000 samples thus far and have had this technology around for about a year. MIHC is a sought-after assay that we run quite frequently, but it's an assay that takes quite a long time. So the numbers kind of indicate that. We've ran so far about 872 samples altogether. Genomics is somewhere in between. And then we also have our CyTOF, which is somewhat of a medium throughput assay, and we have the numbers there as well. So a little bit about maybe some of the assays that we offer. I'm going to start off with our serum plasma cytokine assay using the Olink technology. That's called PEA, which stands for Proximity Extension Assay. This is a system that uses antibodies that carry unique DNA tags. Those DNA tags undergo a process of hybridization, and the hybridization product is then addressed, amplified by a qPCR readout. So it's a very accurate readout. We are able to hyperplex up to 96 proteins from 1 microliter of blood, which is an advantage of that technology. Uses very little material. Because of the double linker hybridization step, we're also able to reduce the amount of background noise, so the data and the signal is pretty clear. We offer a number of panels for the Olink technology, including a target 48 protein panel. So that's 48 proteins hyperplexed, and we report that in normalized counts as well as actual numbers. And we also have our target 96 panel, which we'll report as normalized counts. How do we work with PNNL? We actually had just completed our first project that was led by Dr. Liu and Dr. Rodland. We were able to use the Framingham study. We've received 1,833 longitudinal serum samples that were collected from various stages of cancer progression from these patients, starting with pre-diagnosis samples and then post-diagnosis sample, and then pre-metastases and post-metastases. So we ran 2 panels for this project. The first one was a target 96 panel in immuno-oncology, and then we also ran the inflammation target 96 panel. The samples came to us beautifully randomized, accounting for sex, case control, status, as well as cancer type and site that they were collected from. As a result of that, we have beautiful PCA plots showing that the samples did very well in terms of the normalization strategies that we employed in making sure that the data on such a large number of samples was normalized well. This is just one example. We look forward to seeing the data. I think it's been currently handled by one of the analysts at the PNNL group. Moving on, here's our MIHC platform. So this is in situ profiling of immune contexture. This is the single cell level, iterative cyclic multiplex IHC, so single chromosome staining. We acquired the images after each stain and then computationally superimposed them upon each other and able to segment and quantify with a single cell resolution, the presence of different immune cells and their lineages. Here's an example of our Discovery 23 panel, which delineates very nicely the contexture of most of the immune cells. We also have other panels available that have been validated in multiple cancer types, as well as multiple cancer stages. We have a big immune T-cell functional panel, as well as myeloid panel. We just finished building and validating a big state-of-the-art NK cell-specific panel. So those are all available. I think might be interesting addition as a cross-validation and orthogonal validation for some of the work that you guys do at PNNL side. Just examples of how that data is analyzed, looking at densities per millimeter square, as well as normalized densities. We like to incorporate any clinical metadata, as well as neighborhood analyses. On the right-hand side, you see a Sankey plot, which kind of shows how the different immune cell subtypes will change based upon where they are located, for example, within a tumor, or between samples, for example, between cycle 1 and cycle 2, or pre and post therapy. Some of the things that we work on internally are also, we're really interested in developing an atlas of how the immune contexture changes in different types of cancers, whether there are any patterns that we could see, for example. And here we have a brief overview of some of the cancer types that we work on routinely. And you can kind of see that the immune composition varies between all of those. Some cancer subtypes are very immune poor, like pancreas, for example, and there are some that are super rich, for example, like ovary or even liver. Obviously, the spleen and tonsils are sites of immune infiltration, and they look like they showcase that by including a lot of different immune cells in those types of tissues. Some of the examples how we work with our industry partners, one of the things that I hear a lot about when we deliver our data to our clients is like, well, what does that mean in a context, right? Like, just because you say, hey, there's a lot of this type of cell or that type of cell in our in the immune environment, there isn't really a good context in terms of how that relates to this cancer or any other types of cancer. So entering the field of immunotherapy as part of precision medicine these days, because there are so many different immunotherapies on the market, the company was really interested trying to define what the combinations of checkpoint markers were in this case of metastatic colon cancer. And we're able to go in and with our T-cell functional panel really describe very well to the company what the percentages of the different type of immune checkpoints were in this given cancer and how, more importantly, they combined with each other. So then this is important for potential design of clinical trials, looking at different types of checkpoints expressed at different type of cancer tissue, and then how to match best checkpoint inhibitor or an agonist with the appropriate cancer type. Moving on, just a brief description of our nanostring geomics digital spatial pathology platform. So this is a good complement to our MIHC panel. It is really focused on establishing readouts from phosphoproteins, which MIHC platform doesn't really address very well. This is not a single cell resolution assay, so it is usually read out as quantitative oligos counts per ROI, and ROI is designed as a region of interest that could be changed depending upon the study design. But this specific technology utilizes a UV light linker that could be tagged to different types of tumor masks. For example, we can use PanCK to tag all the immune cells and then pull the signal from different types of phosphoproteins from that region that are CK positive. Some of our panels that we have been able to develop internally, that work is led by Dr. Chris Corliss and Dr. Junho Lee. We have a number of interesting panels that we offer for that assay, including immune cell profiling, but also looking at different signaling pathways, including cell death, PI3 kinase, AKT pathway, as well as custom OHSU developed panel, one focusing on DNA damage and repair, and one focusing on describing tumor microenvironment, as well as other different perturbations, as well as MAP kinase signaling. So very interesting. It took a very long time to develop these, especially with the use of phosphomarkers, which are not always easy to work with. Another assay that we run at IMCO is CyTOF. So this assay is performed in blood cells, PBMCs, or whole blood, and it is allowing us to self-phenotype different types of immune cells in the periphery. Here I'm showing two panels. We have our CyTOF panel 1 that gives us a very nice breakdown of all the different cell phenotypes present in the blood. And then in red here, we see all the functional markers that we then overlay on top of the cell phenotypes to be able to give some functionality, informed functionality to these cell types and what they do and how they're behaving. In the bottom here, you can see the changes in the immune composition from baseline to post-treatment A and then post-treatment B, and you can kind of follow along and see how the different subpopulations of immune cells will contract and expand as a reflection of the treatments. And last but not least, this is our cyclic immunofluorescence platform. This is another spatial biology platform that we have recently deployed. As I had mentioned a little bit earlier, the multiplex immunohistochemistry assay, as much as it is tremendously useful and it really allows us for deep integration of the interrogation of the immune system in situ, it does take pretty long time. Typical turnaround times with analysis and processing are about 8 to 12 weeks. This platform does a lot of automation of what the MIHC platform can do. So it's more of a plug and play it performs really well where you can stain 20 markers on 20 samples in 5 days. So that really shortens our time in terms of from when we get the samples to when we can actually show you data, which is probably going to end up being about 4 weeks from start to finish. So quite a big advancement from our MIHC platform. Another important thing to think about, and the reason why we chose to invest in this technology, is because it's very hands-off. So there's a microfluidic chip that is designed as part of this workflow that allows for all the antibodies and all the buffers to be completely hands-off. And I think we're also are really interested in moving that technology in the future into the CLIA diagnostic space, so we can we can implement some of our discoveries that we make in the research space into patient care. And this particular machine would be very well suited for that because it is very highly automated and requires very little human interaction from that perspective. Again, we have our Discovery Cyclic IF panel that's very similar to the MIHC panel, and we're working on future panels that we're going to be adding into the workflows in the near future, and currently performing validation in different tumor types to really bring this up to speed for some of our upcoming projects. So how do we work with collaborators? Fortunately enough, OHSU and PNNL have a great agreement already in place. In addition to that, IMCO has a special agreement with PNNL as well, where we're able to move projects very quickly, building addendums into existing contracts. So really trying to cut down on the amount of paperwork and legal documentation to move projects swiftly and also be able to pivot if necessary. We get involved in sponsored research agreements as well as grants. Last fall, I was fortunate enough to have been able to write a grant with Dr. Tom Matz from PNNL. Unfortunately, that grant did not score as well as we thought it was going to be, but nevertheless, I do want to say it was a great experience working with the PNNL scientists and being able to put this together. As I previously had mentioned, we also do a variety of other things, helping with development of SOPs and really taking assays that are developed in the research space and help our clinicians as well as our researchers to scale them appropriately so we can access them quickly and get the data out in an appropriate time. I'm going to be around for the rest of the day today. I would love to have conversations if any of this has sparked any interest, and feel free to also email me at my personal email or mcall@ohsu.edu. Thank you for your attention. And of course, most importantly, here's part of my fearless team with my leader, Dr. Corliss, who couldn't be here today, but is currently traveling. So I'll stop there and take any questions if there are any questions.
Great. Thank you so much, everyone. I want to thank our outgoing PMedIC directors and our incoming PMedIC directors, and both Joanna and John, for their talks. If the two co-presenters, Joanna and John, want to come out to the stage, and then if we could get Jamie and Josh up to the stage. If any of our audience would like to ask a question, please raise your hand and I can hand you the mic. And we can then take some questions off of our virtual attendees as well. So any questions to start out?
Thank you. I wanted to thank all the speakers for the great overview. I'm Christy Antonio, and I'm the scientific director of Brian Drucker's lab, but I've been working with PNNL on a U01 funded grant on AML. So mainly, Joanna, I was interested in your Lunaphore technology and if those panels can be customizable at all.
Yes.
Yeah.
So one of the great advantages of cyclic IF platforms, especially the Lunaphore, is that about 85% of our single chromogen antibodies translate really well into the fluorescent. We also have done some preliminary work showing that all kinds of tissue types, even tissue types that generally don't do well with the harsher environment of the MIHC cyclic chromogenic staining, do actually do much better in terms of the tissue preservation. And we are also able to incorporate, because of that, we're also able to incorporate up to 40 markers in a single panel. So any specific targets that you might be interested in, if there is a primary antibody that you know of that already works, there's a pretty good chance that we'll be able to validate it in the fluorescent space as well.
I think one of our goals for the UO1 is really response to drug treatment in patients, specifically clinical trial samples. And then, we have obviously, would want to validate anything you would find out from proteomics, again, in a different technology, and being able to do that in samples, say, in our case, it would be blood clots or fine needle biopsies. That would be helpful to do, giving a more specific panel or something.
Yeah, absolutely. Yeah, and it's a little bit faster than the traditional MIHC, and it does preserve the spatial component as well. Mm-hmm. We haven't tried it with the fluorescent panel, with the fluorescent instrument, but we have data showing that blood clots as well as bone marrow do very well through the process. So I would not anticipate any issues with that type of analysis. We'd be very excited to try it.
Okay, any other questions from the audience, in-person audience?
I'll follow that question while, Liz, you're walking over. Is that platform also, have you tried it in other species or just humans so far? Because I was very impressed by it.
Right. So one of the caveats of IMCO, I think, that I hope to address in the future is the fact that we have been pretty human focused, and cancer focused. And I would really like to change both of those things in the upcoming future. I'm a neuroscientist by training, actually, and sort of came onto the cancer field just by interests and a little bit of luck and a little bit of life circumstances. So my goal really is to broaden our test options to other species as well as other contexts and other biologies aside from cancer. So I was just telling someone while we were eating lunch, I don't hardly ever think about monkeys, but I would love to think about them.
I would love for you to think about them too.
For sure, because it's a great model and it's so powerful. So I'd love to have a conversation about that as well.
Hi. Thanks, the panel. But I want to ask some questions to John Jacobs regarding the mass spec. So you mentioned that you have this very cool technology where you add deuterated peptides into the mixture of the samples and be able to specifically quantify. So my question is that how is the sensitivity of that assay in terms of, for example, you have a plasma sample or serum or urine samples, and how good, and even I just want to kind of really get an idea in terms of not necessarily the size of the volume of the samples, but the sensitivity of the assay.
Specificity, yeah. So each time, let's say you want a multiplex, or you've got however many peptides or targets you're wanting to go after. I mean, there's an assay development approach that goes towards that. It starts out. So to straight out answer your question, most of the time we can develop an assay that is very specific for that specific peptide, with the caveat that, number one, we've been able to identify a peptide that is unique and is detectable and is specific for what you're looking for, whether it's just to quantify the protein or whether to quantify a piece of that protein or wherever, and it uniquely quantifies that protein. But throughout that process, that's basically what our development is working towards, is that, you know, we have a lot of approaches in order to find a good peptide, whether it's in our own data or whether it's in someone else's data or whether it's in you know, in vitro data, or I mean, in silico data. And we don't even have to have seen that peptide before. We can go and look at a sequence and go, this would be a very good peptide. We have algorithms that tell us whether the peptide might be a good peptide. Maybe the sensitivity hasn't been good enough in our global studies, but we want to go looking for that peptide. That's fine. We do that all the time. And we would identify that peptide. We go, once we and then if we get a good synthesis, we get a good synthesized heavy label peptide. That heavy label peptide is our marker, and we can follow that marker throughout all of our chromatography, separations, and we know exactly where that peptide, that heavy label peptide is, and then we go look for the endogenous peak, and we have enough sensitivity and enough different platforms that increase our sensitivity over time that eventually we will find that endogenous peak if it's there. And because we have that marker there, that marker peptide, and we're just linking it to what we're able to find, with enough separation, with enough sensitivity that we're able to detect that. So with some caveats, we're generally pretty confident that if you've got a target, we can be very specific, see that for that sequence for your target. So hope that answers your question. You bet.
Great.
My question is for Joanna. That's a great array of technologies that you have in the IMCO. How do you go about integrating more than one platform on the same sample? Do you do that? What are the limitations? How many different technologies can you integrate? And do you do it by alternative slices on an FFPE block, or how do you do that?
It's a great question. Thank you. So I guess I'll start by saying that most of our assays try to be tissue sparing. So the DSP platform, as well as the MIHC platform, as well as the cyclic IF platform, just use one single section of FFPE tissue. We have developed methodologies that allows us to register the images by using hematoxylin. So we try not to have a sequential H&E stain unless there is a reason for it. So from that perspective, we are usually able to run at least 3 assays on less than 5 micrometer sections. So with Olink, as you may know, that's 1 microliter of either serum or plasma, or in some other cases, cell media, and we've done all kinds of things with it. It's hard to pipette, so the reality is a little more like a 10 microliter or 15 maybe, since it is kind of plexed and requires a multi-channel pipette. We have not tried to combine proteomics with spatial transcriptomics after, but I do think that that's coming. The current technologies are, I hope there are no sponsors listening, but are slightly underperforming at the moment in that realm as far as my opinion goes. But I think that we will get there pretty soon. We're going to be able to achieve the coupling of transcriptome and proteome on the same slide.
Because obviously where I'm going is, can we combine John's laser capture in 5 micron diameter sections with your multi-IHC or something spatial like that and get, the regions of interest in the full proteome rather than 96.
Yeah, for sure. I think there's, that would be incredible.
Great. I would like to maybe give some of our virtual attendees a moment too. Is there anyone online that would like to come off mute and you can introduce yourself and please ask a question? Okay, well, we'll go ahead and hand this back to Christina.
I just had more of a question about the OHSU grants and whether or not they're going to be having a couple of those larger grants next year. And another follow-up question about the internship program that has been established and how are people selected for that, and you know, how can we identify students that might be involved in that, or do they have to be already involved with PMedIC grants to be able to be eligible for that?
I'll take that. We won't put Jamie on the spot 10 minutes into her new job. In terms of the budget, in terms of the money, there's the hope is that there will be funds this coming year. I don't think that the budget is completely finalized for next year, but the request has certainly gone in. These are considered, you know, just in general, there's been a pretty high return on investment for these collaborations. So they are looked upon pretty positively by the powers that be. In terms of the internship, any grad student at OHSU who has, you know, would benefit from working at PNNL for a while is welcome to go do that. There's not a selection process for that. The main thing is their mentor needs to know first before they come to us. It has happened the other way around, does not work as well. And particularly, it needs to be sorted out. Are they going to do something that's related to their thesis work where there's maybe a method or something they're going to learn at PNNL? In that case, they can just go to PNNL at whatever agreed upon time. They're still a student at OHSU. They're paid through OHSU. They're getting their research credit for doing their research, and total flexibility to go do that as long as their mentor and somebody at PNNL, you know, is on board and they organize that. If they are doing something that is not related to their thesis work, again, their mentor has to approve of this, but it would be just like any other internship where they go do something at a company for a quarter or something where it's not really part of their research project. In that case, they take a leave of absence from OHSU and then they can go work at PNNL. Now, PNNL, unlike some of the companies where our students go, does not have a thing where they provide health insurance for interns. So the students need to understand if they do that route, they're not going to have health insurance during that quarter. In our experience, we are often far more concerned about that than the students are. Nevertheless, that is, so it's a slightly different process if they want to do something unrelated to their research. And again, their mentor has to agree to that, and there has to be somebody at PNNL. Otherwise, there's no limit on how many people can do this. And it's actually helpful. All of the training grants now for NIGMS are required to offer students opportunities like internships. So it's kind of a win-win. So typically internships have been for a quarter, but there's no rule that has to be what it is. So particularly if somebody's doing some experiments where they need to go to PNNL and do experiments for their thesis, then, you know, I think it just depends on how long that takes.
Hi there. My question is for Dr. Jacobs again, similar to Fikadu. The question's in regards to I think you were talking about nanopots and sort of nanosample. What can be what's the protein detection ability of that? What kind of depth with that? Can you tell me more about that, please?
Yeah.
Thank you.
It's somewhat related to the tissue itself. That's why I didn't really kick out too many numbers because, I mean, we look at FFPE tissue versus flash frozen tissue versus liver tissue versus, muscle tissue versus whatever. So generally you're talking about in the range of several hundred to several thousand proteins being able to be detected, from, say, looking at the LCM tissue from 50 micrometer up to 200 micrometer squared or diameter. I mean, it just really varies between what the tissue is. And generally, we try to get you. We're aiming towards at least 1,000 proteins being quantified. I mean, I've seen with liver tissue with fairly small isolations. I mean, you're getting to a couple thousand, proteins quantified. Yeah, it's very sample specific, and we try to get a benchmark of where we think we should be, or what the expectation might be, what we've seen globally, and then how we want to scale that down and how we feel like we should be able to, the depth of coverage we need to be getting. Like I said, we've got multiple platforms to kind of get higher sensitivity. It comes at a cost of throughput, but the idea is I would say that we're going to probably get you the biggest coverage or the deepest coverage that is going to be possible at that time for that analysis, for that tissue. I don't know if there's many other folks around that do this type of nanoscale type analysis, and so.
Great, thanks.
Sarah,
I think we
go
have
ahead.
time maybe for one more question. I'm not sure, we haven't had any virtual attendee questions. So if there's anybody that is online virtually that wants to unmute and ask a question, please feel free to do so. If not, if there's any other questions here.
I just wanted to follow up a little bit on John's point.
Yes, absolutely.
I think something that's also important to think about is while copy number per cell is really important, when you're starting to break down into single cell, or you're going into a more narrow region, you're starting to understand the phenotype much better. So you don't need 20,000 proteins, which we never see 20,000 proteins. But if you're looking at 2,000 proteins, you're now starting to understand that better. And when you're making a comparison between one condition versus another, 2,000 proteins may be super critical for trying to understand those changes in a way that's much more specific. So, in some cases, we're looking at fewer proteins, but much higher quality.
So the last thing I'll add is that on the PMedIC website, there's a presentation by Ying Zhu and Peter Bar-Gillespie. And Ying gives a very good presentation and application of that technology.
And we also have James Fulcher here, who's...
Yeah, I don't know why I just didn't hand it off to him. My fault.
All right, I think that wraps it up today. I want to thank you all for attending and for everyone attending online, and thank you so much.
Integrated is like
lot.